Hello, everyone. Okay, so we should now be live. Hopefully you can all see and hear us. So I'm going to be moderating this very exciting session. Just going to confirm if anyone wants to write in the chat box that you can hear and see me okay. Okay. Great. So I'm excited to introduce Pedro Bordalo at University of Oxford, who's going to be telling us about memory and representativeness. So I'll let you share your screen and take it away.
Thank you very much.
Great, and we can see your slides.
Everything's working? Great, thanks.
Yes.
So thanks to the organizers for having a paper on the program. This is very exciting for my co-authors and me. So we are all economists. We've recently been working on, well, actually not so recently, starting to work on memory. We think that memory is going to be very important for progress in economics and in finance. And so we're very excited to be here and to interact with all of you. So let me tell you one reason why we think memory is so important. Because we think it can tell us something about the nature of the distortions in beliefs, of belief distortions. Now, specifically, we're very interested in the representativeness heuristic. As you all know, the representativeness heuristic is a tendency to inflate the probability of features of events that are more representative of a group, or sometimes referred to as more similar to a group. For example, people tend to exaggerate the frequency of red-haired Irish, even though the true frequency is quite low. Sometimes people will say that more than half of the Irish have red hair, when in fact, you know, more than most Irish people have actually brown hair. So why does this occur? Well, brown hair might be more likely in the population of the Irish, but red hair is somehow more similar, more representative to the Irish population. Okay, and so we want to understand how this occurs. Now, just a quick note saying that this idea of representativeness-based belief distortions has been making headways in economics and in finance. There's a lot of empirical evidence that it explains, particularly the version of representativeness that models as relative likelihood. So, for example, representativeness helps us understand and describe social stereotypes, beliefs, distorted beliefs about social groups, but it can also be used to better understand expectations in the context of finance, of macroeconomics, and so this, in fact, has been shown to be useful to understand things like booms and busts in economic cycles, for example. So, understanding representativeness is actually something, you know, we would say of first-order importance going forward in economics and in finance. And so the question becomes, why does representativeness work? Where does it come from? Why do people judge likely events or features that are merely representative of or similar to a group? Okay, this is the question we try to address here. The approach we take is that this bias, this representativeness, reflects the selective memory. There's limited and selective memory. So going back to the Irish example, when you say somebody's Irish, you selectively retrieve from memory instances of red-haired Irish people, and that leads you to overestimate the share of Irish people who, in fact, have red hair. So there's a link between selective retrieval, selective recall, and probabilistic assessments. So that's where we're going here. Okay. So what does the paper do? It presents a model of probabilistic judgments based on selective memory. So it's again going through this. It's based on a cued recall model that features similarity and interference. And then it tests, the paper tests the model's predictions in lab experiments and online experiments, okay? So we want to see this paper as one step in a broader agenda, which tries to understand distortions in probabilistic judgments, which are pretty important also in economics and finance. We try to understand that on the basis of its cognitive foundations. Now today, in the interest of time, I will just present a sketch of the model very briefly and spend most of the time talking about the lab experiments and the results of that. Okay. So here's a sketch of the model. There's a decision maker, there's a participant in a lab experiment, and what do they have to do? They have to assess the likelihood of different hypotheses, which we'll index by H, in light of some data D. So for example, I tell you that somebody's Irish, that's the data D is equal to Irish, and what you have to assess is what are the shares of people with different colors of hair. So what's the share of red hair, of Irish that have red hair, for example. That's the hypothesis H. So how do we think of this? We think of this data, this D, as a cue that helps you retrieve a past hypothesis from memory. So D is Irish, retrieves red hair, also potentially brown hair, and so on. But this retrieval, this recall, is biased by representativeness. And so going back to the definition, hypotheses that are more similar to the data will come more easily to mind. That's what we mean by representativeness. And so the key issue here is how do we think of and how do we define the similarity between data, like D equals Irish, and hypothesis, which is, for example, red hair or brown hair. So the key feature in this idea of similarity is contextual interference. So let me explain what this means. So a hypothesis H, so for example, red hair, is perceived to be more similar to the data D, is more easily recalled given the cue of the data D, when H is more likely to occur within the data D. So for example, if there are more red hair people among the Irish, when I tell you that somebody is Irish, you're more likely to retrieve a red hair person. Okay, this is a very kind of non-controversial, I think, hypothesis about recall, so frequency, is shared by many models. The key feature of our approach is the next one, that a given hypothesis is more easily recalled when I give data D if that hypothesis is less likely to occur with a different data minus D. Okay, so the reason that red hair is easy to retrieve when I give you Irish is not only because there are relatively many red-haired people among the Irish, but also because there are very, very few red-haired people among different nationalities, different data. So instead of D equals Irish, different data, for example, other Europeans. Conversely, why is it hard to think about a brown-haired Irish when I give you D equals to Irish? Because other groups, other data, like Southern Europeans, for example, have such a large share of brown-haired, dark-haired people. So that hypothesis, H equals brown hair, is very common across different data, and so it's hard to retrieve when I give you the D equals Irish. So this is what we mean by contextual interference. There's interference coming from the hypothesis via its impact on its relationship to other data minus D. Okay, so this is going to be the sketch of the model. We'll come back to it briefly towards the end. So we're building on a large body of research about memory. We're specifically thinking about cue recall, where a cue, here we're thinking about the data, retrieves memories from the database that subject to the forces of similarity and interference. So items that are more similar cause less similar items to be neglected. The key innovation of our approach is that there's another form, a new form of interference, which in a sense flows back from the items, from the hypothesis you're thinking about to the data. So this is the idea that it's hard to think about a brown-haired Irish person because there's so many brown hair amongst different data that you should not be thinking about, but they interfere with the retrieval of the brown-haired Irish person. Okay. Our work is also related to a large body of research thinking about the role of memory and probabilistic judgments. There's a number of the models that we discuss in detail in the paper in the interest of time, I will not discuss them most here, but just to say that this feature, that the relationship between a hypothesis like red hair and other data that you're not being curious about, shaping your probabilistic assessments about the data that you're given, does not arise in these other models. So this context dependence, that your beliefs about a given group, say the Irish, depend on the distributions of other groups, does not arise. It's kind of specific to this hypothesis of contextual interference. So let me now describe to you an experiment, a first experiment that seeks to test the key prediction of contextual interference of our model. I'll go through 3 experiments in total. So but they all have the same broad design. So in this experiment, participants see a sequence of 50 images. They come one by one. I'll show you an example in a second. These 50 images include 25 numbers and 25 decoy images. That could be either words or shapes, depending on the treatment. And each image, or each number, or each decoy image has a color. It could be blue or orange or gray. And subjects are told that after seeing this sequence, they will be asked questions about the images. Okay? So let me give you a brief example of a part of a treatment. So participants see a sequence of 50 images, just like the ones I just showed you. The ones I showed you were words and numbers of different colors, blue and orange. And after they've seen the 50 images, they are asked, the computer randomly chose one image from the ones that were shown to you. The chosen image is a number. What is the likely color of the chosen image? Is it blue or is it orange? So you are given a cue, or this piece of data. There is an image that is a number, and you have to make a probabilistic assessment about the distribution of colors. Is it blue? Is it more likely to be blue or orange? Okay. So how do we test this? I mean, this paradigm is clearly involves memory and retrieval. But how do we test the idea of contextual interference? Well, we have 2 treatments. In both treatments, numbers, the 25 numbers, have the same distribution. So there are 10 orange numbers and 15 blue numbers in both treatments. What changes across treatments is what are the other images that people see. In the control treatment, you see 25 gray shapes. And in the blue words treatment, you see 25 blue words. Now, what is the idea of this experiment? Participants are always asked about their assessments, the probabilistic assessments about the distribution of numbers, and that's constant across treatments. What varies is what other groups they see, what are the decoys that they see, and if their assessments about numbers change when you replace gray shapes with blue words, then that's evidence in favor of contextual interference, because the hypothesis of, because there among the decoys, there are blue words in the second treatment, okay, which will interfere with the retrieval of blue numbers, but there's no such interference in the control treatment. All right. So when you're asked about colors, the prediction is, sorry, there's my mouse. Can't see it. The prediction is that in the blue words treatment, here we go. In the blue words treatment, the presence of the blue words will make it harder to retrieve the blue numbers when you're thinking about the numbers, and therefore will lead to the inflation of the probability of orange numbers. Okay? And so here is, we have actually a number of output measures. We ask about the likely color, that's what we just seen. We also ask about the probability of different colors directly, and we ask about how many orange and blue numbers did you see, so we recall all instances. This is a graph about the results. So what you see is that in the right-hand side, you see there is the share of people who say that the number is more likely to be orange is about 35% when in the gray treatment, and it jumps to 55% in the blue treatment. So people are much more likely to say that numbers are likely to be orange when numbers are presented alongside blue words. Okay, again, consistent with the hypothesis that the presence of blue words interferes with the recall of blue numbers. It makes orange more similar to numbers more similar to orange. Of course, this result holds across all the different output measures that we look at. So the likely color, the probability, the numbers of orange numbers recalled, and so on. Now, if it is true that what is driving these results, let's see here, what's driving these results is the fact that blue words make it harder for you to think about blue numbers when you're assessing the probability distribution of numbers, then it should be the case that by reducing the number of blue words and perhaps adding some orange numbers will reduce the treatment effect between having no interference, like in the gray shapes, and having the treatment effect. Okay? And the reason is that if you add some orange words, then there is some interference for blue numbers, but also some interference for orange numbers. Okay, and these might balance out. And so the distortions about the numbers will be smaller if you add some orange words. Okay, and so the treatment effect relative to the control will be small. Okay. Now, so what we do is that we, in fact, replace some of the blue words by orange words, and we see what happens to the share of people who say, when they are asked about what's the likely color of the number, what happens to the share of people who say that it is orange, and that share of people goes down. Okay, and you can see here the results, both from online experiments and also from lab experiments. And there's a clear decrease of the share of people who say that the words are that the numbers are orange. And if you increase the share of orange words, that effect is kind of gradual and becomes stronger and stronger as you go. And in fact, as we add more and more, let's see as you add more and more orange words, the results become closer and closer to the results when you don't have any blue or orange words at all. Just have gray shapes here on the right-hand side.
And you have about 2 minutes left.
2 minutes left. Okay, sorry. So this suggests that there's interference in recall. Let me say two things. What we do is we have this model where we have a functional form for interference, for similarity between hypotheses like colors and data like numbers. This similarity relationship builds on the Tversky similarity relationship. So it's a similarity relationship that is context dependent. It depends, the similarity between orange and numbers depends on what happens with words. And we actually calibrate the model and see it performs quite well. And this context dependence factor is very important. Let me just conclude by talking about a third experiment, which really tries to get at the idea of cued recall. And the idea is that depending, if you have different cues, then you can have different processes of recall and different processes of contextual interference, which might shape your probabilistic assessments. So how do we test this? We have all participants see the exact same images, okay? We just change the cue afterwards, the question that they have to answer. And the images are as follows. There are still 10 orange numbers and 15 blue numbers, and there are 25 blue words as before. But now they can be large or small. So if I show you all these images and I ask you, a number was drawn, what's its color? Then we are in this case, just as like we were before. And here there's a lot of interference from the blue words, dampening the recall of blue numbers, such that you inflate the probability of orange numbers. Okay? But instead, if I ask you, you just saw all these images, a number was drawn, what's the likely size? Is it large or small? Then we designed this set of numbers in a way that all the blue words are large and all the orange numbers are large. And in fact, the blue numbers are small. So if I ask you about the size, here the interference happens across the large hypothesis. There's no interference for the small hypothesis. And so what you get is that if I ask you about the size, you will inflate the probability of small numbers, which are blue. If I ask you about the color, you'll inflate the probability of orange numbers, which are large. So just by changing the Q, I can change the way you think. I can change the similarity between the data and the hypothesis and what you retrieve and the probabilistic assessments. And this graph shows you that as predicted by the model, if I ask you for the color Q, you inflate the probability of orange. If I ask you for the size Q, you in fact lower the probability of orange. So the results go through for all the output measures. So just to conclude, what we take out of the paper is that it's very suggestive that we form beliefs, probabilistic judgments and beliefs, by retrieving information from memory. This retrieval is selective. It's based on similarity, and we have this important idea of similarity that has a contextual interference in it. And this helps explain a range of things. The observation that judgments about probability and about similarity go hand in hand. It helps us understand why some unlikely events, like the red-haired Irish, are systematically overestimated. It helps us understand why beliefs are context dependent. And so we hope that understanding better how memory works in this specific type of problems helps us understand belief distortions more generally. Thank you very much.
Thank you. So I guess now we will invite up Jessica Wachter from the Wharton School of Business, and we'll save questions once she's done with her talk to be the discussant for what we just saw. Oh, I think you're still muted.
Oh, okay.
Oh, great. Now I can hear you.
Okay. Thank you. Hi, everyone. Thank you to the organizers for inviting me to discuss this paper. And now I will start sharing my screen. There's my discussion. Okay. So I hope that you can see, there it is, my discussion. Okay. All right. So I really enjoyed reading this paper. Everything look okay? Thumbs up? Thanks. Okay. So here we go. So the motivation of this paper is how do people make decisions under uncertainty? And so one idea, not the only one, but one idea is that they make probabilistic judgments. So they consider hypothetical outcomes and attach a probability to each hypothetical outcome. Of course, that requires estimating a probability of the outcome. And Kahneman and Tversky, in a series of papers, present evidence that people make large errors in judging the probabilities. So what types of errors do people make? Well, it turns out they can make really basic errors. So if the truth is if A, then B, well, the type of errors people make is they think if B, then A. So for example, if the truth is if somebody has a disease, then the test is positive. Kahneman and Tversky present evidence that instead doctors think if the test is positive, then people have a disease, which if the disease is rare can be very wrong. In the red-haired Irish example, which personally it never fails to get me every time, if red hair then Irish is the truth, I mean, not literally, but almost true, people tend to think if somebody's Irish, they must have red hair, but in fact, it's very rare to have red hair, and most Irish people of Irish descent, have brown hair. Okay, so one issue with these results is that perhaps these come from longstanding semantic associations, and that's what Sudeep Bhatia talks about. But the nice thing about this experiment is it strips away from that, and so we're going to look at a setting where there's really carefully get away from these semantic associations. Okay, so here's the experiment. I'm only going to talk about the first one. in the interest of time. So the gray treatment, participants see 10 orange numbers, 15 blue numbers, 25 gray shapes. And you saw how this worked. In the blue treatment, participants see 10 orange numbers, 15 blue numbers, and 25 blue words. Participants are then asked, after seeing all of these things flash across the screen, if the image is a number, what color is most likely? Okay, so gray treatment is 10 orange numbers, 15 blue numbers, 25 gray shapes. The blue treatment is 10 orange numbers, 15 blue numbers, and 25 blue words. I find it helpful to think in terms of these contingency matrices where across the columns is the type of shape and across the rows is the color. So in the gray treatment, half of the images that are shown are gray shapes, and then the numbers are divided among blue and orange, with the majority being blue. So the correct answer to the question, by the way, is blue. Among the blue treatment, there's half of the shapes are blue words, and then the remainder are divided between blue and orange numbers. Okay, so those are the contingencies, the correct contingencies. Okay, so the results. So remember, participants are asked, if the image is a number, what color is most likely? So the participants in blue incorrectly tend to report orange. Participants in gray tend to report blue, though that's less clear. The main thing is that the difference is statistically significant between blue and gray. So if in terms of the A then B example that I had before, it's like if orange then number, participants in blue say if number then orange. So if orange, yeah, then it's definitely a number. That's true. But the participants in blue think, well, if they see a number, that's the cue, well, then it must be orange. Now, of course, it's not quite so easy than that, as that participants in gray do not get confused. If number, then they say blue. Of course, I'm oversimplifying a little bit, but in the interest of fixing ideas. So it's kind of neat that the blue words here cause the confusion, because if you think about what the participants are being asked, they see a number, what color is it? They report orange. Well, where are the blue words? They're nowhere in the question, but it's the presence of the blue words that are messing things up. Okay, so that's the result. So let's talk about the model for a few minutes. So the agents in the model, the agents assess the likelihood of a hypothesis H in light of data D, which is also called the Q. So data Q are the same thing. And the idea here is that hypotheses are similar to the data, that hypotheses that are similar more easily come to mind. Of course, what does similar mean? What does come to mind mean? So we're going to be specific about that. So agents recall H if H is likely to occur with D. Well, that's pretty standard. Agents recall H if H is unlikely to occur with alternative data minus D. Well, that's not so standard. Okay, so the similarity here, similarity between hypothesis and data, is the probability that H and D occur together, kind of the contingencies, minus some coefficient, we'll call it gamma, times the probability that H occurs with minus D. So the idea here is that if H occurs a lot with minus D, that reduces the similarity. And then you apply the loose choice rule to tell you what's the probability that the agent's going to tell you H. And it's not actually entirely clear whether this is a model of an agent's subjective probability of recalling H, or is it actually, is it a construct for fitting probabilistic data? But let's leave that question aside for a moment. What I want to highlight here for a moment is that this is clearly a relative measure of similarity, right? Because you can make any two things appear not very similar potentially by throwing in a lot of minus Ds. And then you can kind of just reduce a S that way. Let me just give you an illustration of why I'm saying that. So I'll give you an illustration through the experiment itself. So let's first think about the blue case. Well, in the blue case, this makes eminent sense. It's obvious as what's going on. So you're assessing the similarity between orange and number. Well, it's high. Why is it high? Well, orange is likely to occur with numbers and unlikely to occur with words. I mean, there's no orange words, right? On the other hand, blue is likely to occur with numbers, and it's likely to occur with words. So this is a way of bringing in the blue words. Remember, that's like the whole problem here. And, poor blue here is getting penalized through this term because it appears with words. Okay, so that's how this works. Now let's think about, though, how it works in the gray treatment. So in the gray treatment, okay, similarity between orange and number is high. It's likely to occur with numbers. It's unlikely to occur with shapes. Blue is likely to occur with numbers. It's also unlikely to occur with shapes. So now participants are trading off the small differences, presumably, in PhD instead of PH minus D. And gray is unlikely to occur with numbers and likely to occur with shapes. Okay, so agents, so orange or blue are going to appear much more likely than gray. Nobody's going to say gray. And blue is kind of going to win out because, you know, PhD is somewhat higher for blue. That's what's going on with gray. Okay, so here's kind of my concern. So my concern, as suggested before, is what's this mysterious minus D? You know, what's not the data? And what are the H primes? And what I like about this experiment is that actually, while in the Kahneman-Tversky examples, it's sort of kind of constructed so that minus D and H are sort of obvious, or H prime. It's actually a little less obvious in this experiment, and that makes the experiment more like real life. So ideally, agents should compare H and D across all possible Ds and all possible Hs. But think about what's going on in the gray treatment. Return here. So what's this agent doing? Is this agent assessing all 6 of these contingencies? Or is the agent just asking, likely to occur with numbers, likely to occur with numbers? The agent doesn't care, shouldn't care, about the shapes, and shouldn't care about gray at all. And in fact, of course, when you do the math, these things drop out. But it matters if you're thinking about an explanation for what is the agent doing. So the question is, when we write down the similarity model, how do we choose what's included in minus D? And how do we choose what are the other hypotheses that the agent is considering? Is the agent considering minus D to mean shapes? Is the agent considering the other hypotheses to mean gray? You know, and maybe we can consider other Ds and other Hs. Maybe words should be included in the hypotheses. Maybe Ds should also include like lilac or, you know, other colors. Maybe faces should be included. So how do we know what to conclude and what include and what not? And of course, the more things we include, then the more impossible this becomes. But if we don't include things, then we can't really use this. So it's a little hard to know where to draw the line. It's much more computationally intensive than standard Bayesian decision making. Now, of course, as I said, in the loose choice rule, truly irrelevant things drop out, but you don't know that ex ante. So I guess what I would suggest is, in some ways, a more intuitive model is one where, when asked about numbers in the gray treatment, so think about an agent who's just gone, a participant who's just gone through the gray treatment. Maybe the agent doesn't think about shapes or words at all. This participant just thinks about numbers and colors. Okay. So
we could wrap up in about a minute or so.
Okay. All right. So in the spirit of this, I'm going to, in 1 minute, present an alternative model. So this is actually in ongoing work with Michael Kahana. Okay, so if you think about these contingency matrices that I introduced at the beginning, we can perhaps think about subjective contingency matrices as arising from associations of context and features, where contexts come from colors and features come from the type of image. Okay, so how could this model work? Well, obviously, if it's going to work, you would need something like features numbers to retrieve context orange. So you need a lot of context oranges and features numbers to appear together. Well, actually, that you can get something like that in a iterative process of contextual retrieval, encoding, and features retrieval. And the reason for that is, if you look back at these matrices and consider what's going to make you think about a number. Well, if you're in the blue treatment, only orange is going to make you think about a number. So if you're in kind of an orange context, then orange is going to lead you to think about a number, and that's going to cement an orange number association. A word is going to lead you to think about blue. Well, that's already associated. In the gray treatment, numbers are going to, either blue or orange context are going to lead you into numbers. So you won't get the same bias. So I believe that a model where you have retrieval of context can essentially introduce a bias where you get more orange number pairs than blue number pairs in the agent's matrix. Okay, so I will wrap up. So I really love this paper, I will say. I think that the experimental results demonstrate that representativeness is indeed a fundamental phenomenon, which makes it really interesting to people like me, who I'm an economist, and so I have to kind of pick and choose what, how I'm going to model agents' thinking. I think that the similarity-based model that the authors have is parsimonious and easy to work with, and that this paper opens pathways, new pathways in ways of thinking about representativeness and decision-making more broadly. Thanks.
Thank you so much for an interesting talk and interesting discussion. So I guess, Pedro, if you want to respond to anything that Jessica raised, we'll wait to see if anyone else has any questions.
Hi. Thanks, Jessica, for a great discussion, very clear discussion of what we do. I think the question of what H is, as what the H's are, and what the minus D is very important. It's something we're thinking about. I think one way to think about that, but again, we try to have a parsimonious approach. But one way to think about that is when I ask you about numbers, you try to think about some numbers. They could be orange or blue. And once you get to blue, it's very easy through kind of a similarity jump, I think it's in a way similar to what into the sequential sampling that you were thinking about. You think about blue, you start thinking about words, and you remember words, and that's when you remember words, you say, okay, I'm supposed to think about numbers. I have to stop and go back to numbers. So it might be there might be a way to endogenize what the minus D is, if it's connected to the data, to the queue, through the exploration of the types. So if you think about blue, you might go end up in a different data set because there's no overlap. So nothing reminds you of gray, and so you never go to the gray shapes there. And so this is one way in which kind of the problem endogenously kind of generates what's the minus D. And the same thing with H. In principle, H should be, you should think about all the possibilities, right? I think there's evidence. Here it's very simple. I mean, here we try to keep it very simple. It has just 2 colors, maybe 3 colors. If I ask about colors, it's easy for you you just saw them like a few minutes ago. It's easy for you to remember the set of colors. And in fact, the cube color should remind you of the set of colors. But in other cases, you might, if you have many hypotheses, then you might forget some. Like in the disjunction fallacy, that's what happens. You kind of you start thinking about some hypotheses and you neglect some. Yeah, and so that's another interesting thing. How far do you go? Which hypotheses might you might neglect? And that should all be possible to think about in the context of a similarity relation. And I think the context is a very important part of this. Here we try to keep context, very simple. You just in an experiment, we assume that everything is the same context, and you're thinking about the experiment recently. But it's generically very important aspect. So thanks a lot for the questions. These are future work.
Yeah, thank you. I think both between not having any further questions and in the interest of time, we'll end things there. But thanks again to both of you.
Thank you.
And so for our next talk, we'll be hearing from Wei Tang from Indiana University at Bloomington, discussing reinstatement of temporal context observed with human scalp EEG during successful episodic memory retrieval. Hello.
Hello.
Oh, hi, Lynn. Can you hear me?
Hi. Yeah, I can hear you.
Hi, everybody.
Great. So if you want to try sharing your slides. Great. Can see your slides. So
Okay, great.
Away.
Hi, everyone. I'm Wei, and I want to first thank the CEMS committee for letting me presenting this work. It's my first CEMS, and I've been having a great time. So this is an EEG study on the reinstatement of temporal context observed during episodic memory retrieval. And this work is in collaboration with Mark Howard and neuroimager He-Shen Liu, who collected the data. So I'm interested in the mental jump back in time phenomenon. This is a concept in episodic memory to describe the experience when a memory is successfully retrieved. We also recover a previous state of temporal context. For example, say a person experiences a series of events, A, B, C, D, and so on, and later they encounters event C again. So this will trigger a mental jump back in time for them to re-experience C as from the past, but they are also likely to remember events B and A, which was in the temporal context of C when it happened. So cognitive models for this phenomenon can treat temporal context as a slowly decaying signal through time during memory encoding. At the time of retrieval, not only the representation of the event is reactivated, but also the temporal context is reactivated together. This temporal context model, or TCM, can explain the behavioral contiguity effect, such that items that are encoded closer in time are likely to be recalled together. And this contiguity effect can be measured by a lag CRP curve, which describes the higher transition probability of recall for items that are next to each other on a steady list than items that are further apart. The TCM predicts that the neural correlates of temporal context is also a slowly changing signal through time. So when we compare the neural signal during retrieval versus the signal during encoding, we should see the highest similarity between the reinstated signal with its own representation in the past, which will show as a high similarity at lag 0. In addition, because of the slowly changing temporal context during encoding, the reinstatement signal will also be similar to neighboring items during encoding. So this similarity as a function of lag will resemble a lag CRP curve as in behavior, and we call it a neural contiguity effect. So the neural contiguity effect has been observed with invasive measurement in the human brain, such as single unit recordings and the ECOG signal in epileptic patients. This encouraging evidence supported the existence of a temporal context signal in the human brain. Now, the question is, can we measure it with a non-invasive method, such as EEG, which is widely applicable to the healthy population and also different, say, development and aging groups, so we can study memory in a wider population? And the major obstacle of analyzing EEG signal is that it contains very high background noise. So this prominent background noise caused by neurophysiological fluctuations that is not directly associated with task, it will bury the reinstatement signal of temporal context in the high amplitude of noise. And this noise is also a slowly changing signal. So no matter what kind of task you're applying, you will always find high autocorrelation in the EEG signal due to the noise. because of this noisy autocorrelation, any contiguity analysis, the similarity will be contaminated by the noisy autocorrelation. And this is the main problem we are trying to address in this project. So we used a standard old-new recognition task. In the study session, 100 words were shown to the subjects, and 20 minutes later, in the test session, the same 100 words were mixed with 100 new words and shown to the subjects in a random order. And the subjects were asked to report whether each word was seen in the study session or whether it was new. And we took the test signal for our analysis. So the hypothesis is that if temporal context is successfully retrieved, then things being equal, the reinstated temporal context will cause the words that are neighboring to each other in the study list to have higher similarity than words that are further apart in the study list. In other words, we should be able to recover the lags between words on the study list just using the reinstated signal in the test session. However, in the test session, things are not equal in that this background noise autocorrelation will make the words that are closer in the test session to be highly correlated. And to mitigate this noise correlation, we took a difference of the retrieval signal between each old word and its neighboring new words. The assumption is that reinstatement signal is only contained in the old words, but not the new words. So the difference will give us this reinstatement signal plus a bunch of residuals. The residuals are the difference of the noise. If we take the difference of each old word and its neighboring new word and look at the correlation between the difference signal, then we'll see that because of if the temporal context existed in the reinstated signal, then they should be correlated. And the residuals of the noise are unlikely to be correlated. So our plan is to apply the similarity analysis between these different signals. So we took an additional step to decompose the EEG signal into spectral domain. So we have multiple time frequency components. For each time frequency component, we make the difference between old and new words. And there were 70 channels. So the difference will be a 70-channel feature vector. For each old word, we obtain this feature vector. And the similarity between the vectors are measured by normalized inner products, which will result in a scalar value that ranges from -1 and 1. And this is our similarity score. And we measure this score between each old words at different lags. And because there are multiple time frequency components, at the end, for each lag, we are getting a matrix that is cross frequency at time to represent the similarity between words. So the hypothesis is that if temporal context is recovered, then these similarity matrices will show gradual changes as a function of lag. And here is the result we are getting. When looking at the similarity matrices averaged across subjects and trials, we see at the global level, there is a difference between shorter lag and longer lags. At lags 1 and 2, the overall level of similarity is greater than those at lags 3 and longer. This suggests that there might be contiguity effect, and maybe it's stronger in certain time frequency components than others. So to locate which time frequency components have a stronger contiguity effect, we measured the slope of similarity across lags for each component. The assumption is that a more negative slope will represent a stronger contiguity effect. So this matrix shows the slope at each time frequency component. So the black contours shows the 95 percentile from a permutation test. So the permutation test pointed to two significant clusters that has strong contiguity effects. One cluster is an early component following stimulus onset, and it is in the gamma frequency range between 30 to 40 hertz. And another component is a late theta component centering around 600 milliseconds after stimulus onset. And just to verify there is a contiguity effect, we plotted out the average similarity scores from these two clusters as a function of lag. And we can see there is a gradual decay. And lastly, we tested the correlation between the slope and reaction time. And there is a significant positive correlation suggesting that subjects with stronger contiguity effect will respond faster.
And you have about a minute until the, oh, great. Okay.
Yeah, so altogether, we observed that items encoded closer in time will have similar neural activity during retrieval. And this mechanism can be observed with scalp EEG. So it opened the door to non-invasive studies of jump back in time in episodic memory. And so there are further steps we're considering to dissociating what we observed as a contiguity effect with possible confound like correlated encoding. And we would like to locate the sources where the contiguity effect comes from with the simultaneously recorded MEG or fMRI signals. And finally, we would also like to test on subjects from different development aging groups and asking different questions about this temporal context signal. And last, I want to thank all the wonderful people that contributed to this work, and thank you for your attention.
Great, thank you. So I guess I'll just give it a minute if people want to add their questions to the add the question box. We have a few minutes for questions. And if there aren't any questions, oh, I see there's one question from Eitan Schechtman. Would high-pass filtering, instead of computing difference between neighbors, also work for eliminating slow drifts?
Yeah, that's a great question. So taking the difference is a type of high-pass filtering in theory. So it's applied a lot in time series analysis as exactly a high-pass filtering to denoise. So basically, they're the same thing.
If there aren't any other questions, I might ask, I mean, I guess it's a little bit different with your data, but I'm also trying to think about how to translate this into thinking about forward versus backward lags, just because I know that for the backwards lags, that can be somewhat important in free recall for distinguishing more of an item reinstatement from a context reinstatement. And if you had ways about thinking about that order or maybe something at encoding. Sorry, and that could be our last question.
I confess I'm new to memory models, and my background is in neuroimaging. So based on my conversation with Mark and Zoran, what I understand is that the neural signal tend to not have an asymmetry in the contiguity effect. That's the difference from behavior. And the exact reason why I don't think I haven't drilled down to but that's as what I know as a matter of theory or fact, that the neural contiguity effect is symmetric. So
right. If anything, I think capturing that symmetry with the backwards lags is more what I think will be critical for future steps. But
okay, I see. Yeah, thank you for the suggestion. Yeah, I'll look more into it.
Thank you. Okay, so Up next, we have Sebastian Michelmann from Princeton University, who will be talking about one-shot learning of naturalistic story and how that improves predictions on a fast time scale in the auditory cortex.
Okay. Hi. Can you?
Yes.
Okay. Okay. So I'm gonna try sharing my screen.
Sounds good. I'll let you know if I can see it.
Okay. Screen one. Okay. And do you see my presenter view or the real view? Okay.
I can see the real view, just the title slide. Oh, now it went back to.
Yeah, close it again.
Okay.
Okay, should I just start presenting?
Yes, please. Thank you.
Okay. Hi, everybody. I'm Sebastian. I'm a postdoc at Princeton with Ken Norman and Ulrich Hassan. And today I want to talk to you about how the tracking of learning during naturalistic story listening can help us to reveal underlying interactions between hippocampus and cortex. So when I was a child, our parents used to tell us stories. And when a story had ended, my siblings and I would often ask our parents to tell it again. And I distinctly remember how much I loved that on the second time of listening, I could already tell what was about to happen. And I was also looking forward to the best parts of the story and emotionally preparing for the scary ones. So when we study learning during such naturalistic conditions, we know that this type of learning is possible in one shot. And we can also observe here naturalistic prediction. That is, after learning, we can predict upcoming moments in the story from memory. And importantly, when we investigate learning naturalistically, there's another benefit. We can study the role of event boundaries in the encoding and retrieval of information. And from the work of many of you, we also have a clear hypothesis here that encoding of information should take place at event boundaries in the story. So what I want to show you today are some highlights from a study in which we investigate learning when participants are listening to the same story twice. The whole study actually contains data from participants that perform boundary norming and prediction experiments on mTurk. But today, I really want to focus on results from 9 ECOG patients that are undergoing pre-surgical monitoring for clinical purposes as they listen to the same story twice. And the temporal resolution of ECOG, together with the spatial specificity, has the potential here to inform us on a fine-grained temporal scale how processes during encoding and retrieval are orchestrated between different structures. And just to mention again, the mTurk data here is only important when I talk today about event boundaries in the story, because we derive them from these human annotators. So the first thing that I want to show you is how we can identify neural prediction learning when patients are listening to the same story twice. And to do this, we use Granger causality in a very unconventional way. We apply the math of Granger causality that is typically used to assess connectivity between channels to the data at each channel, comparing the first and the second run of listening. Specifically, we take the high-frequency gamma power at run 1 and at run 2 of listening and ask if the signal acquires additional predictive information on the second run that was not there on the first run. So Granger causality makes use of regressive models across time. Therefore, at a given electrode, we will get an autoregressive model predicting each signal from its own past. Now, we can additionally then predict every time point from the previous signal at the other run. And the logic here is that if there is predictive signal that emerges from memory, then these previous time points in the second run should help to improve the prediction on the first run, but this should not work the other way around. So contrasting the two, we see significant evidence for predictive recall on the second run of listening in the auditory cortex. And because Granger causality uses regressive models, we can actually make a prediction now based on these models and see where the model predicts the data at every moment in the story. And from this, we learn when predictive recall takes place in the story. And what I'm showing you now is a video of when in the story run 2 accurately improves the prediction of run 1, that is, when predictive recall takes place. So whenever this line here goes up, this is the moment that was predicted from memory on the second run of listening. And I just want to show you a brief segment now. I'm going to play this video and you will listen to the story and just focus on what happens at this peak here when the brain signal tells us there is additional predictive information that emerges from memory.
I began my illustrious career in journalism in the Bronx, where I toiled as a hard-boiled reporter for the RAM.
Yeah, so this makes sense. The beginning of the story after music is something that you wouldn't anticipate on the first run of listening. However, on the second run, we have memory to help us to predict this onset. And just to mention other moments in the story that are highly predicted in neural data are, for example, moments of profanity, when the main character is introduced, and all kinds of other moments. So we now have channels where we observe significant predictive recall in the cortex, here plotted in blue. But we know that episodic memory also hinges on the hippocampus as a key structure, which is plotted here in red. And crucially, so what we would expect at this point is that at some point during the story, while participants are listening, information needs to flow between cortex and hippocampus in order to encode and retrieve the information that subserves predictive recall here on these cortical channels. Crucially, we hypothesize that the encoding of information from cortical channels in the hippocampus should happen at event boundaries. And to speak to this, we do a connectivity analysis where we assess multivariate mutual information between cortex and hippocampus, and we can do this analysis at different lags. So we could have information come from hippocampus and go to cortex, and this would be a hippocampal lead, or we could have information in cortex that then flows to hippocampus, which would express itself in a hippocampal lag. And finally, we can do this whole analysis at every time point across event boundaries, therefore giving us a 2D map of shared information between the structures. And I'm conditioning this analysis on the 0 lag, which is typically spurious. Now here I'm showing you this connectivity profile between hippocampus and cortex on the first run of listening. And you see on the x-axis hippocampal lead to lag, which is the shift between the two structures, and then you can see the distance to the event boundary on the y-axis. And the color here codes the amount of shared information between the two structures. And crucially, we observe a cluster of increased shared information peaking at a hippocampal lag of approximately 300 milliseconds, so information flowing from cortex to hippocampus, and this takes place approximately 700 milliseconds prior to this event boundary, which is marked by the button press of the individual participants on mTurk. And now to do a control analysis, we can look at the visual cortex, and here we do not observe such an increased information flow. And this difference is also a significant one. Now we can repeat this whole analysis again on the second run of listening, and here we find even stronger information flow from cortex to hippocampus that also starts substantially earlier. And again, when we look in the visual control region, we do not observe this shared information here between cortex and the structure. So, however, what I've shown you now is information flow from cortex to hippocampus. But in order to perform predictive recall, we would also need, at some point, information to flow from hippocampus to cortex. So arguably, these human annotated event boundaries are maybe not the best or the most sensitive handle, however, to this predictive recall. However, luckily we have this time course of predictive recall for every moment in the story. And we have these interesting peaks here where we can observe strong and correct predictive recall in the neural data. And importantly, we also have this data for every subject. And just on a side note, if we log this time course of predictive recall to event boundaries, we also observe increased predictive recall in the vicinity of event boundaries. So there is a link between predictive recall and event boundaries in this data. What I want to show you now on my last slide is, again, the information flow between cortical electrodes and hippocampus. However, we are now locking these moments to these moments of predictive recall. So we have a one-dimensional plot showing the shared information between hippocampus and cortex at different lags, again, starting on the left with hippocampus leading information flow from hippocampus to cortex, and on the right, information flow from cortex to hippocampus around these peak moments of predictive recall. And what we see on the first run of listening is that there is no real increased connectivity between the structures that exceeds information on shared information with other channels, or what we obtain from phase shuffled random data. On the second run of listening, however, we do observe increased connectivity with the hippocampal lead now. So information flows from hippocampus to cortex prior to these moments of predictive recall. And then this is also followed by 2 moments of information flow from cortex to hippocampus. So in conclusion, I hope I could show you that we can track predictive recall in auditory cortex by hijacking the measure of Granger causality for our purposes. And during the continuous narrative, information flows from cortex to hippocampus at event boundaries, and this is very much in line with this idea of encoding of information or storing of snapshots at the event boundaries. And finally, prior to moments of strong and correct predictive recall, information flows from hippocampus to auditory cortex, and this is followed by reverse information flow. Then I want to thank everybody in Princeton, in the Hassan lab and the Norman lab. First and foremost, of course, Ken Norman and Uri Hassan, my 2 supervisors. I also want to highlight Adine Flinker's group at NYU, who are doing the ECOG data collection, and Amy Price, who is a former postdoc in the Hassan lab, who collected this data. And of course, I want to thank all of you for listening and the organizers for this opportunity to speak here today.
Great, thank you so much. So I guess we'll turn to questions, and it looks like there's 1 question. So now there are 2, but starting with the one that has the most votes. So Richard Atkinson, or it might be Richard, I guess, would a change in story in story 2 that surprises the subject produce a larger predictive signal than the one you have observed?
So the way our analysis works is basically we can only make use of information that is shared between the stories, because we use information on the second time of listening that is predictive of what is about to happen on the first time of listening. I mean, it's an interesting question, like how surprise would play into this if we now started to violate predictions and see what happens in the neural signal. But we can't really speak to this from this data, unfortunately.
Thanks. So another question that got a lot of votes is from Anna Shapiro. She says, really exciting work. Do you think this relates to the cortex to hippocampus to cortex flow of information that's seen during endogenous replay?
Yes, I do very much think so. So I think what we see is reinstatement of information in sensory areas, which I don't think is surprising, but and I think like this is a similar mechanism, like where I know replay is. I'm assuming Anna means replay now in the terms of at rest, because there's like this distinction of replay of reinstatement of information in general, or replay reinstatement of information during rest or during sleep. But I think the structures and mechanisms, in my opinion, should be the same, and I think we could also potentially use this method to address replay that happens during in other situations than just this predictive recall during repeated story listening.
So thank you. So I'll ask one more question, which is from Ryan Hubbard. He says, it seems possible that aspects of the narrative could influence the prediction signals. Do you think you could track the predictability of the narrative using language models such as GPT and relate this to the neural signals you observe?
That's a very good question, and a very accurate question, because other people are doing this. Actually in our lab at Princeton, and this is in the Hassan lab, where they are trying to just look into general language prediction that is not dependent on episodic memory. And yes, you can. I don't know how much I can spoil here, but they are also looking into GPT-2 and this language other language prediction models. So yeah, so I think that's very much on point that you could track this, but in terms of just general natural language processing, you wouldn't need the second repetition, because we don't really the reason why we have it, or what it brings to the table here, is that we can distinguish what is predicted automatically, which is captured in the autoregressive model of range of causality, which contrasted with what is only predicted more in the second run. So we can really see what is the prediction gain that we get from having heard this story before, so that we can actually use episodic memory to improve predictions. So this is like the focus of this project. But yeah, very much other projects are looking into this natural language prediction, and it's also something where you would use predictive models to track brain signals in ECOG.
Great, thank you so much. No, I think it's always an exciting answer to a question if someone thinks of something and there's yes, we've already thought about it, we're doing it.
Yeah, I'm not doing it.
That other people are thinking about it also. Thank you. So I think with that, we'll thank you again for your time and move on to next talk. So finally, to end this session, we have Alexandra Cohen from New York University, who will be talking to us about influences of reward motivation on behavioral and neural memory processes across age. And great, I can see your slides. So if you want to get started.
Okay, great. Thank you so much to the organizers for a fantastic conference and for the opportunity to share some of my work. The promise of potential reward can help us to prioritize information and memory. For example, a student may be more motivated to remember material that their teacher highlighted as being important when they're preparing to try and ace a test. Studies that have been primarily conducted in adult animals and humans have shown that reward-motivated encoding enhances memory, typically after a delay. In adults, we see that memory for high-reward memoranda is related to increased activity and connectivity of the mesolimbic dopamine system, centered around the ventral tegmental area and the hippocampus, both during and after encoding. Post-encoding interactions between the VTA and category-selective visual cortex involved in processing high-reward memoranda have also been linked with better high-reward associative memory, which is thought to reflect a reward-motivated memory consolidation mechanism. Additionally, interactions between the VTA and lateral prefrontal cortex have been associated with reward-motivated behaviors and reward-related memory formation. More generally, we know that dopamine-dependent plasticity in the hippocampus is critical for memory formation, which may have unique implications during adolescence. Although little is known about how reward motivation in the formation of lasting memories influences the formation of lasting memories across development, converging evidence from studies in rodents and postmortem studies in humans suggests that dopamine signaling in subcortical brain areas peaks during adolescence, which may lead to particularly strong memory representations of rewarding events relative to more mundane events. To test this possibility, we asked, how do reward associations influence memory across age? And are there age-related changes in underlying post-encoding consolidation processes involving the mesolimbic dopamine system? We hypothesized that reward associations might have a larger influence on memory during adolescence due to increased mesolimbic dopamine system connectivity. To address these questions, we collected data from 90 participants, ages 8 to 25, in a 2-day fMRI and behavioral study. Although for some of the visualizations we refer to age groups, all analyses treated age as a continuous variable. So first, I'll go ahead and walk you through the experimental procedures which were adapted from fMRI tasks used to study reward-motivated memory processes in adults. On the first day, participants completed the fMRI study, and their first scan was just a baseline active rest or arrow scan, where they were simply asked to push a button to indicate in which direction an arrow was pointing. Next, they completed a localizer, which I'll describe more in a moment, and then they completed reward-motivated encoding. So prior to entering the scanner, participants were told that they should make up stories about the 2 images that they saw on the screen in order to help them remember that they went together. They were told to pay special attention to the color background that the images were on, because gold images meant that remembering those images went together would help them win a big bonus, and silver backgrounds meant that remembering those images went together would help them win a small bonus. On the left-hand side of the screen, participants saw trial unique objects, which were selected ahead of time to be familiar to individuals across ages. And on the right-hand side of the screen, they saw one of 8 different source images. And these source images were either faces or places. So for half of our participants, faces were the high reward category, and for the other half, places were the high reward category. And this was the activity we were looking for in the localizer, subject-specific face or place activation. After that, they underwent a post-encoding active rest or arrows period, followed by retrieval, where they saw half of the images, trial unique images they had just seen, and they were asked to recall which source image went with that object. They did this all again, and then 24 hours later, they returned to the lab where they completed a retrieval for all of the paired associates that they had seen the day prior. We designed this task so that we would be able to address a number of questions about age-related changes in motivated memory processes. And in line with our primary hypothesis, we first focused on asking, how do reward associations influence memory across age? We examined associative memories retrieved 24 hours after encoding, which included both paired associates that had been retrieved on day 1, as well as paired associates that had not been retrieved. So here I'm plotting for you the proportion of associative hits by age, reward level, high or low, and whether the paired associates had been retrieved or not on day 1. We found that while memory performance improves with age, we see facilitation of memory for high reward associations relative to low reward associations across age. And although there is substantial forgetting of paired associates that were not retrieved on day 1, we observed reward motivated memory enhancements regardless of whether or not the paired associates were or were not retrieved. We next assessed whether, in parallel to the behavioral results, post-encoding consolidation processes that might facilitate the prioritization of high reward associations in memory were also similar across age. In other words, we asked, are there age-related differences in post-encoding VTA functional connectivity? We were still interested in possible age-related differences because past studies of reward-motivated behaviors in both humans and rodents have shown differences in mesolimbic dopamine system activity in adolescents relative to adults in the absence of differences in behavior. We addressed this possibility by examining seed-based changes in the arrows or active rest phases of the task following reward-motivated encoding relative to the baseline across age. Specifically, we took our arrows runs, and for each of them, we first regressed out the brain activity related to the task. We then extracted signal from the VTA and performed a seed-based functional connectivity analysis. We examined experience-dependent changes in whole brain functional connectivity, as well as with high reward category selective cortex, given prior work suggesting a role for category selective visual cortex in the formation of associative memories. So turning first to the whole brain analysis, consistent with prior work, we found significantly increased post-encoding connectivity between the VTA and bilateral hippocampus across all of our participants, as well as increased VTA connectivity with several other brain areas. We next examined whether changes in connectivity varied with age. And while we did not find changes in connectivity that increased with age, we did find changes in connectivity that decreased with age. Specifically, we found one cluster with peak activation in the lateral prefrontal cortex, which has been previously implicated in driving reward-motivated behaviors, that showed decreasing VTA connectivity with age. To understand how this increase in experience-dependent connectivity that is particularly elevated in our younger participants might relate to motivated memory, we performed regressions examining the relationship between high reward associative memory performance, connectivity, and age, controlling for the high reward category of source images. Surprisingly, we found that the relationship between post-encoding VTA lateral PFC connectivity, as well as VTA high reward category selective cortex connectivity and high reward associative memory after 24 hours varied with age. In our older participants, increased post-encoding connectivity was associated with better high reward associative memory. And this relationship between the VTA and high reward category selective cortex connectivity and high reward associative memory in our young adults mirrors the findings of Deepu Murthy and colleagues in just adults. We didn't observe a relationship between changes in connectivity and high reward associative memory in adolescents, And younger participants actually seemed to show the opposite pattern, such that increased connectivity was associated with worse memory performance. This indicated that the particularly increased connectivity we observed between the VTA and lateral PFC in our youngest participants was actually associated with worse high reward memory. Both of these relationships were specific to the associations that had been retrieved on day 1 and remain statistically significant when controlling for low reward associative memory performance. The VTA doesn't simply show this connectivity behavior relationship across the whole brain, as connectivity with a paracingulate region that comes out of the whole brain age contrast, and also with low reward category selective cortex do not show this relationship. In summary, and returning to our key questions, we found that reward motivation enhances memory with age from childhood to adulthood. And despite observing similar reward-motivated memory facilitation effects across age, we found that memory enhancements may be differentially supported by connectivity of the mesolimbic dopamine system across age, such that while increased post-encoding connectivity between the VTA, lateral prefrontal cortex, and high-reward category selective cortex is associated with better high-reward associative memory in young adults, it's actually associated with worse high-reward associative memory in children. And this result might align with findings from Bita Mokadam's group that demonstrate differences in how reward-motivated events are encoded by VTA neurons in adolescent relative to adult rats. Still, this leaves open questions as to what brain mechanisms support reward-motivated memories in our younger participants, and whether adolescence might be a time when these mechanisms are in flux. We hope to address these questions, and we also plan to conduct multivariate analyses to investigate how reward motivation influences memory representations across age. And with that, I'd like to thank my advisor, Kate Hartley, our collaborator, Leila Davachi, all of my fantastic RAs who collected the data for this study, participants and their families, and our funding sources. And thank all of you for your attention.
Great, thank you. So I guess we'll give it a minute if anyone wants to put a question into the Ask a Question box. We have a few minutes for questions. And while we're waiting for that, I'll add that I say we have a few minutes for questions, but I realize we're running a little bit behind. So we'll plan to start the next session at 2:10 p.m. So running a little bit later than originally planned. I guess we'll give it just another minute if anyone has any questions. Well, I guess I'll ask you, admittedly, this is a little bit more on the threshold of what I know, so I apologize, but I guess if it's a simple question, it'll be easier to answer. But how have you thought about this in relationship to, I guess, how different types of rewards might impact the memory, and that depending on age, that might influence how they're thinking about what it means to have that reward, if that makes sense. It might not necessarily be, if it's the influence of reward based off of just how the reward contributes to their memory, as opposed to just a more objective measure of reward, which I realize is, it's easier for me to say sitting this versus having a good experimental manipulation to get at that. But
yeah, I mean, I think in terms of reward motivated behaviors more broadly in the developmental world, there seem to be sort of similar effects, whether you're talking about a social reward or a monetary reward or something else. I think it would be interesting to consider specifically how that might affect the types of results we're seeing, but I don't have any strong intuitions for how things would differ.
Thanks. And now I see there are a couple of questions showing up. So I'll ask you a question by Michael Cohen. What do you think you'd find if it were less associative or semantic?
Yeah, so we do have the item level data from day 2 as well, and there do seem to be these same reward motivated memory enhancements for items. We did particularly want to target associative memory in this task, but But I think given the nature of our design, we can look and see how these processes change as a function of these different types of memory. But I'm sure that if we were just looking at individual items, we might see something different. But I would have to think about that a little harder. Thanks for the question.
Thanks. So I'll end by asking you one other question that's here by Jiahu Po, who says, great talk. And is there a possibility that the difference in reward effect across age is driven by increased use of strategy in adults?
Yeah, that's an interesting question. We do have some self-report data afterwards to try and get at the types of strategies that people were using, if any, and there doesn't seem to be, I haven't looked at it in a minute, so I'll check and get back to you. But at least the last time I looked at it, there didn't seem to be any sort of consistent strategies that people were using that varied across age. But we do have some data to try and look into that a little bit more specifically.
Interesting. Well, thank you so much. And so that wraps up this session of Virtual CEMS, and we will resume here at 2:10.